Good afternoon, everybody. Welcome to a special Friday seminar sponsored by the Center for Evolutionary Medicine. Quick news for those interested in the center in particular. We have great news that Ben Trumbull has signed today, and he will be joining us in a year. He's the person who has samples from the Chimani and all kinds of projects about relationships of health in modern and pre-modern environments. Today's talk will be by Susanne Pfeifer, and we're so glad to have her here. Her topic, as you see, is the importance of mutation and recombination variation rate, lessons from chimpanzees and lizards, following up on a number of similar projects, including a very large paper in Science a few years ago looking at balancing selection in humans and chimpanzees. She took her undergraduate degree in Germany, where she's from. Are you from someplace close to Sauerbrunn?
Yes, very close, but very tiny.
Yeah, and that was actually in the Max Planck Institute for Computer Sciences, where she took her undergraduate degree. She stayed there in the Center for Bioinformatics and did a Master's of Science project on identification of molecular markers in the Arabidopsis gene. And then she went to Oxford, where she took her degree in statistics with Gilman Bain. She then went to Vienna and did a postdoc. That was in the Center for Integrative Bioinformatics. And then she's been back in Switzerland, or in Switzerland, more recently. This work covers a wide variety of areas, but they're all tied together, I think, as you'll see, very tightly. She also has had multiple awards for travel and other kinds of things to get her around to present these results, and a number of other publications. She'll summarize some of them in this talk. So glad you're here, Susan.
Thank you. Thank you very much for the invitation and the kind introduction. It's a great pleasure for me to be here today and to talk to you about some of my main research interests and some of the work I've been doing over the last few years. As we all know, molecular evolution is driven by an interplay of different forces: mutation, recombination, selection, genetic drift, and migration. Out of these, mutation is often regarded as the raw material of evolution, simply because it constantly gives rise to new variation in genomes, making it the primary source of genetic diversity within populations and genetic divergence between species. Ultimately, mutations underlie all evolutionary adaptations and also all heritable diseases. And thus, it's really interesting to learn more about the understanding of the mechanisms by which they arise and estimate their rate of current occurrence. The rate of mutation is mainly controlled by two opposing forces: the benefits of reducing deleterious mutations, which impose a fitness cost on the carrying individual, and the costs of increasing fidelity and accuracy. Mutation rates are known to vary widely between different species, between different populations, individuals, and sexes. For instance, in primates, we know that males exhibit higher point mutation rates than females, which is a subject that Melissa here is actively working on. But even on a regional scale, within a single chromosome, we can see a very large variation between different chromosomes, but also if we look within a single chromosome, both on the megabase and kilobase scale, as you can see in the picture here, whereby the largest variation that we observe is actually seen at the smallest scale at individual sites of the genome. Although different genetic factors have been identified that influence mutation rates at different genomic scales, very much about the rate variation remains unexplained, and more complex processes are likely involved that are currently very poorly understood. Thus, I think the study of mutation variation and the forces driving it is of great importance, because it enables us to gain a better understanding about many biological phenomena and also evolutionary processes. In fact, I would argue it is one of the main goals of evolutionary and population genetics, simply because so many key questions rely on the accurate information about genome-wide mutation rates. For example, if we think about comparative and functional genomics, we need an accurate estimation of mutation rates if we want to date and infer and learn about demographic events inferred from genetic data, because the number of mutations that we see between different chromosomes provide us with a record of the time that has elapsed since the most recent common ancestor. Knowledge about mutation rates is also important when we try and define a null model, when we try and infer selection, but also in medical genetics, which I probably don't need to convince you about. Knowledge about mutation rates is essential because mutations have severe impacts on human health by causing genetic diseases. Of equal importance, I think, is the study of recombination, which is a fundamental process in sexually reproducing species, which plays a very critical role in the production of viable gametes. Mechanistically, chiasmata generate a physical connection between homologous chromosomes during meiosis that is required for proper segregation. Thus, in the absence of a backup mechanism, which is the case in humans, at least one crossover per chromosome is required in every meiosis in order to ensure that there's a proper disjunction. What we know is that too few crossovers in humans can lead to aneuploidy, which is highly deleterious. Autosomal monosomy is embryonic lethal, and embryos with trisomies either do not survive full term or they have really severe developmental disabilities. On the other hand, illegitimate ectopic recombination between homologous but non-allelic stretches of DNA are a very important source of mutation, and the outcomes have been implicated in many genetic disorders. But in addition to ensuring the correct segregation of homologous chromosomes during meiosis, recombination is also a very important evolutionary process because it shuffles existing genetic variation into new combinations on which natural selection can then act on, helping species to adapt maybe more quickly to environmental challenges. Given the fundamental importance of recombination, we would probably think that this process is very tightly regulated. And in some aspects, it clearly is. There are numerous mechanisms, for instance, in humans working that ensure that there's really one recombination happening per chromosome in every single meiosis. But on the other hand, we also see that recombination is tremendously variable. We see rate variation differences among species, populations, sexes, but also within single individuals at virtually every scale that we examined, at single loci up to entire whole genomes. So this raises really important questions about the recombination rate variation and its determinants, such as which aspects of recombination rate are under genetic control, how many loci are involved, are the same loci contributing to both broad and fine-scale recombination rate variation that we observe, and to what extent are these effects sex-specific. Trying to find answers to some of these questions was what captured my interest first in studying population genetics, and at the time I started my undergrad in Germany, as Randy just mentioned, the first human genome had just been published. The HapMap project was in its very early stages, and the first high-throughput sequencing technologies were just on their way. The prospect of a so far unseen wealth of data that was enabling us to obtain a better understanding or better insight into the distribution of genetic variation within and between individuals, populations, and species also meant that we needed much more efficient data analysis because the amount of data that was around imposed really severe statistical and computational challenges. And this is why I decided to study computational molecular biology, which is a mix of traditional computer science, statistics, and biology. For my undergrads, so both my bachelor's and master's, I studied at Saarland University, which is a small university in Germany, but one out of only 5 that back in the days offered proper bioinformatic courses, and at the Max Planck Institute for Computer Science, where I was developing methods for identifying molecular markers in genomes for my honors thesis. I then moved to the University of Oxford, where I was working on the statistical challenges of detecting mutation and variation from high-throughput sequencing data in the group of Gail McQueen, with direct applications in primate population genetics in a collaboration between Molly Perschwowski's group, back then at the University of Chicago, and Peter Donnelly's group at the Wellcome Trust Center for Human Genetics. I continued in the area of primate population genomics in Vienna, together with Arne von Haesler and Magnus Nordborg. And earlier this year, I moved to Lausanne in Switzerland, where I'm currently a visiting researcher in Jeff Jensen's lab, and where I'm mainly collaborating with Hopi Hoekstra's lab at Harvard and Brie Rosenblum's lab at Berkeley. So in general, I would say I'm interested in analyzing information that is carried by extant variation to learn about genetic and evolutionary processes by combining large-scale data sets and data analysis and modeling. Currently, my 2 main foci are to understand how natural selection shapes patterns of genetic variation in different species, and to elucidate the causes and consequences of recombination rate variation. Thus, today I'll be giving you 3 examples. One studying recombination rate evolution in primates, and 2 examples focusing on the evolution of cryptic coloration in wild populations in lizards and deer mice. So although recombination is a very important genetic process, we still understand relatively little about where it occurs and the mechanisms underlying it. And the major limiting factor here is the very low availability of genome-wide fine-scale genetic maps. And just to give you an idea, by the time I started my PhD, detailed recombination maps were only available for humans and the distantly related model species of mouse and yeast. So for a very long time, the main reason for this low availability was that the direct observation of recombination events is a technically really difficult process, limiting its usage and making it impractical for most of the species we're actually interested in. So on the one hand side, we have traditional large-scale pedigree analysis, which have really low resolution practically just because there are a few meiosis that can be observed. On the other hand, we have sperm typing studies, which allow us to investigate many thousands of meiotic products from a single individual, but obviously these studies are male-specific and very often limited to only a few individuals and small regions of the genome. So in the past few years, the statistical analysis of large-scale population genetic data provided us with an interesting alternative means to estimate fine-scale historical and sex-averaged recombination maps, an approach that really has greatly improved our understanding of the process of recombination, especially in humans. However, I think there's a really big problem in using population genetic data, which is that cryptic errors in variant calls are often missed by state-of-the-art variant filters. And importantly, these errors can be observed across all reference sequences, from the draft version of a sequence up until a really high-quality finished reference genome like the human genome. And the underlying error modes really impact all analysis of genetic variation from high-throughput sequencing data. Thus, I think this problem, which is often somewhat neglected, is really affecting the work of every single one of you here working with high-throughput sequencing data. Errors are really inevitable, but I think also for that reason it is really important that we're trying to understand the different types of errors that can occur in our data and that we develop better statistical models that enable us to distinguish real signal from noise. And this is something I've been working on a lot during my PhD, and I continue to actively work on because I think sequencing protocols and sequencing technologies are changing really rapidly. And so we continuously really face new challenges in this area. And I think it's a really important area because both false positive and false negative results can severely alter the results of all of our studies. So just to give you an example, in the work I'm going to talk about next, for estimation of recombination rates, if we think of false positive variant calls, then they really often artificially disrupt haplotype structure, and that obviously leads to wrong estimates. In the interest of time, I will not go into any of the details here, but I rather present you directly with the study. But just very briefly before I'm doing that, I want to say a few things about what was known from different studies of human recombination rates by the time we started the project. So what we knew is that meiotic recombination in humans tend to occur in very short segments of the DNA, about 1 to 2 kilobase long, which are termed recombination hotspots simply because recombination in these regions is much more frequent than in the surrounding sequence. The study of these recombination hotspots has led to the identification of a 13 base pair DNA motif, which is associated with about 40% of all recombination hotspots in humans. And the location of these hotspots is greatly influenced by the zinc finger protein PRDM9, which specifically binds to this DNA motif during meiosis, and which generates histone 3 lysine 4 trimethylation marks, which eventually leads to double-strand breaks and both crossover and non-crossover solutions. Very interestingly, in some species without PRDM9, such as in dogs, yeast, or for instance Arabidopsis, hotspots tend to occur at promoter regions, while in other species they don't have PRDM9, such as Drosophila, honeybees, C. elegans. We don't see any recombination hotspots at all. And currently, we have no idea why that's the case. We have no idea what's the underlying mechanisms. And I think this is one of the reasons why studying recombination rate evolution is so interesting. So in humans, the zinc finger domain of this PRDM9 motif evolves really quickly, with evidence of positive selection on the residues in contact with DNA. And as a result, what we observe is that there is a very rapid turnover of hotspot locations. And given this rapid turnover, you might ask the question whether we can see any hotspot sharing between humans and other great apes, whether they are PRDM9 independent or not. And to answer this question and other related questions, a team of collaborators, who I'll mention at the end of the talk, and I decided to study variation and recombination rate evolution in our evolutionary closest living relative, in chimpanzees, by developing a methodology to construct a fine-scale genetic map from high-throughput sequencing data from 10 Western chimpanzee individuals. So Western chimpanzees are 1 out of 4 subspecies of chimpanzees. The study represents one chapter of my PhD thesis that has led to a joint first author publication. Again, in the interest of time, I will not go into any of the statistical or computational details. I'll rather tell you directly what we've learned from it. So when we have a look at the PROT scale on the chromosomal level, the protist scale, then recombination rates are actually incredibly similar between humans and chimpanzees. As you can see in this graph here, showing you the genome-wide comparison of recombination rate on the y-axis for individual chromosomes arrayed along the x-axis for chimpanzees on top and humans on the bottom. But what you can hopefully also see is that apart from this conserved chromosomal scale, that even at the megabase scale, similarities emerge between chimpanzees and humans, particularly driven by this telomeric rate increase in the recombination rate in the 2 species. Yet, we also found regions of substantial divergence, whereby the most dramatic change in prot-scale recombination rate that we observed occurred when we were comparing the short arms of chromosome 2A and 2B in chimpanzees with the autologous region in humans, which is chromosome 2. So chromosome 2 was created by a telomeric fusion event sometime in the ancestral human lineage. And what you can hopefully see is that in chimpanzee and chromosome 2A in blue here and 2B in red, we see this telomeric rate increase. But then when we look at the fused region in humans, which I show you here in gray, then recombination rates actually have decreased by 3-fold. And I think this is quite nice because in a way nature here provided us with an experiment which allowed us to explore the effect of chromosomal organization on recombination. Interestingly, the degree to which recombination events are concentrated in this fused region is not any different than in the unfused region around it, indicating that this change in prot-scale recombination rates that we observe was not accomplished by specifically eliminating crossover events at hotspots. At the fine scale, the landscape of recombination in chimpanzees is dominated by hotspots to a similar extent than what we see in African populations, as you can see in this plot here, which shows you the concentration of recombination in fine scale maps for chimpanzees, shown here in red, and for human populations. So we have humans of European ancestry in green and humans of African ancestry in blue, which you can't really see very well because it's lying right behind the red line. But what you can hopefully see is that in chimpanzees, about 80% of the recombination happens in only maybe 20% of the sequence, which are our recombination hotspots. But then when we're looking more carefully about recombination rates around hotspots identified in chimpanzees on the left and identified in human populations on the right, what we see is there's no overlap whatsoever. Whenever we see a hotspot in chimpanzees, there's basically a cold spot in humans and vice versa. So there's really no hotspot sharing between the 2 species. Interestingly though, although there's no overlap between any of the hotspot locations, the average fine-scale recombination rates seem similar around some genetic features, such as around genes, which I'm showing you here, where in both species you can see an increase in recombination rate by about 20% around the transcription start site and around the transcription end site, whereas the transcribed region is decreased by about 30%. Such concordance suggests that features that affect chromatin states, such as, for instance, nucleosome occupancy, which is destabilized around promoter regions and CPG islands, may similarly shape the propensity of recombination in these sites in both species. Possibly reflecting a similar effect, we see that recombination rates are increased around CPG islands. The effect is slightly larger in chimpanzees, where recombination rates are increased by about 50% relative to the background rate. As I mentioned earlier, in humans, and as we now know, in some other species as well, a proportion of hotspots contain a binding site for the zinc finger protein PRDM9. When we look at PRDM9 sequences in our Western chimpanzees, we exhibit actually a really great variation, both in the number of zinc fingers, which I show you here as little squares, but also in the identity of DNA residues that bind, whereby I show you here unique combinations of DNA binding residues in different colors. Whereby we see 3 very common alleles indicated here in red of 6 zinc fingers, 16 zinc fingers, and 18 zinc fingers. Computationally predicted DNA binding motifs for these different PRDM9 alleles show a very great overlap in submotifs, which I highlighted here by the colored bars and the tick marks next to the alleles. However, when we actually look at recombination rates, we find no evidence for local increase in recombination rate around any of these predicted binding sites for the PRDM9 variants. In addition, we also can't find another DNA motif that's underlying any of the hotspots that we are seeing. So I think our study raised one really important question, which is why couldn't we observe any hotspot sharing? And I think there are 3 possible explanations for this. The first is that PRDM9 might have lost its role in specifying hotspot locations in humans. However, I don't really think that's the case because we can't find any inactivating mutation. The second possibility is that PRDM9 alleles might have a similar specificity to target DNA sequence, but the substantial allelic diversity, which is much greater than what we see in any of the human populations, may obscure signals for individual alleles. Again, I don't think that is the case because this hypothesis cannot explain why, when both the density and the strength of hotspots on the population level is similar between chimpanzees and African populations, as we've seen earlier, why we can recover known PRDM9 motifs in African human populations, but we can't find a comparable motif in chimpanzees. The third possibility is that PRDM9 might play a similar role than in humans, but individual PRDM9 alleles might simply bind to a much greater variety of target sequences. Unfortunately, I can't give you the answer to which of these hypotheses might be true, because we simply don't know. But I think that makes it clear that further work is definitely needed trying to answer this and other related questions that remained unanswered. And while our study has focused on the comparison of humans and chimpanzees, what we'll ultimately require is information from other primate species in order to further elucidate the role or the factors that affect recombination rate evolution on both the broad and fine scales. Jeff Wald's lab at the university or at UCSF has currently a study under review that looked at recombination rate evolution on a much broader scale, comparing recombination rates in other chimpanzee subspecies, bonobos, and gorillas. And their results suggest that PRDM9 might play an important role more broadly in predicting recombination rate variation across the great apes. So this is obviously in contrast to what we found in our study. So raising the question whether the Western chimpanzees that we studied are somehow unusual in regards to recombination, or whether humans, bonobos, and gorillas are an outlier in terms of the primate clade. And in order to answer this question, what we need is a better understanding of recombination rates in other more distantly related species. So we need an outgroup, but much less work to no work has been done in lesser apes or in old world monkeys. And that's why I used some of my independent research money during my postdoctoral fellowship in Vienna and started to study Vervet monkeys, which are an old world monkey, together with a PhD student who I co-advise. And I think Vervet monkeys are a really interesting subject of research for several reasons. So Vervet monkeys are the most populous African non-human primate. There are thought to be 5 different subspecies, but this is simply based on morphological differences that inhabit most of sub-Saharan Africa. In the 17th and 18th century, Europeans brought, together with the slave trade, a small number of Vervet monkeys to the Caribbean islands of St. Kitts, Nevis, and Barbados, where they started to have feral populations which greatly expanded to a current level of tens of thousands of individuals. So these individuals are demographically comparable to genetically isolated human populations that have emerged from recent bottlenecks and are therefore thought to have a similar advantage in terms of genetic association studies. But apart from that, Vervet monkeys are also among the most important non-human primate models for biomedical research. They are about equally closely related to humans than rhesus macaque, for which there currently is a biomedical or a shortage for biomedical research. And they have been made available for the scientific community for a diverse set of studies, from diabetes to obesity, kind of studying cardiovascular disease or AIDS, which probably Bert is interested in, as the Caribbean-derived monkeys are disease-free in terms of SIV, but they're readily infected with SIV, making them an excellent model system to study the immune response up until initial infection. So as I said, I'm currently working on developing a methodology to construct fine-scale genetic maps from whole sequence, high-throughput sequence data from over 160 wild Vervet monkey individuals, so about 130 wild monkeys from all over sub-Saharan Africa and about 30 more individuals from the Caribbean islands, in order to learn about recombination rate evolution in old world monkeys, but also to learn about potential differences to great apes. Apart from studying recombination rate evolution genome-wide, I'm also very interested in learning about recombination rates within the pseudo-autosomal region. And I think that's really interesting and something we couldn't do in the chimpanzee project, simply due to the very limited sample size that we had there. And I think it's so interesting because the pseudo-autosomal region is a region of true sequence homology between the mammalian X and Y chromosomes, which has undergone really rapid evolution. So in humans, we know that we need at least one crossover per meiosis between the X and Y chromosome in the male meiosis for proper segregation. And this has led in the human populations to really high recombination rates in the pseudo-autosomal regions. They're about 20-fold higher than what we see on the genome-wide average. And again, we really don't know what's the underlying mechanism, what's turning up recombination rates. So I think that would be really interesting to study. But I also think this data set is a really valuable resource for other reasons. I think it's great to get a better understanding of the demographic history of the Vervet monkey and to infer selection, particularly with regards to the coevolution of vervets with SIV, or to get a better understanding of ancient balancing selection with other closely related species, which is another research area I've been working on during my PhD. The second part of my talk will focus on phenotypic adaptation due to recent environmental change. And I think that's really important because the study of populations that have recently colonized new habitats is an area of interest, particularly for evolutionary biology, both because newly encountered environmental pressures can generate strong natural selection, but also because adaptation over very short timescales can be detectable on the genomic level. Over 150 years ago, Charles Darwin still relied on rather intuitive examples to build an argument for his theory, such as the one I quote you here. When we see the red grouse, the color of heather, and the black grouse of peaty earth, we must believe that these tints are of service in preserving them from danger. Perhaps inspired by Darwin's intuition that camouflage confers a selective advantage, and probably also because it's visually so obvious, color variation became a main focus in many of the early studies trying to understand the molecular basis of adaptive evolution. And there are many examples in nature in which cryptic coloration occurs, and it is indeed one of the few examples where the genetic variants underlying adaptation are well understood. So up until today, spontaneous mutations changing coat color have only been reported in 4 genes: the agouti signaling protein, attractin, the melanocortin-1 receptor, in short MC1R, and mahogunin. The protein products of 3 of those interact at the surface of pigment-producing cells, so-called melanocytes, and constitute the machinery that enables the cell to switch between producing dark and light pigments. Thus, mutations in any of these genes can result in a melanic phenotype. Melanism has independently arisen many different times and in many different species, as I just showed you. And in most of the cases studied, mutations in the MC1R gene are responsible for melanism in these natural populations, such as in the case of lizards, which I'm going to talk about first. So what we were interested in particular was to study the phenotypic adaptation of 2 different lizard species to the recently formed white sand system of southern New Mexico, which you can see in the background picture here. And as the name suggests, white sands is a very distinct landscape of white gypsum dunes, with the majority of the sand having been deposited really recently within the last 2,000 to 5,000 years. White sands accommodates a number of different lizard species that have independently and convergently evolved planched forms that live on these sand dunes and that are in really stark contrast to their close relatives that live on the dark soil in the surrounding Chihuahuan desert. Given the young age of these white sand dunes, these populations are expected to be the result of really fairly recent colonizations, and thus the system provides us with a great opportunity to link color variation in wild populations with adaptation to their novel habitats. So the aim of the study, which my colleague Stéphane Laurent and I jointly led, and which just got accepted for publication in Molecular Ecology last week, I think, was to use population-level genomic data to understand the demographic history of natural selection on the genome level for dark and light lizard species, dark and light populations of 2 lizard species. So on the one hand side, we have the little striped whiptail, Aspidoceles innata, and on the other side, we have the southwest fence lizard, Xyloporus cowlesi. And what we already know is that in each of these populations, the blanched coloration is linked to a particular mutation in the MC1R gene. In the case of the little striped whiptail, this mutation prevents receptors from being integrated into the cell membrane. Therefore, no signal is being transmitted to make melanin, whereas in the case of cowlesi, there are simply fewer receptors that are being integrated. Population studies and functional assays could show that there is a different genetic architecture that's underlying these 2 mutations. So the mutation is thought to be dominant in cowlesi, but recessive in innata. And therefore, this system represents one of the very first examples in nature where we can actually directly test differing predictions about recessive and dominant alleles and their hitchhiking effects by studying or comparing the molecular signatures of selection for these alleles in the same gene in the 2 lizard species living in the same environment. Just a few words regarding our study design. So for both target species, we sampled lizard from the 2 contrasting environments at the same locations to enable demographic inference over the same spatial scale. So we sampled both on and off the sand hills. In particular, we sampled 9 individuals from the White Sands National Monument over here, a further 9 individuals from a dark soil population located about 85 kilometers northeast of White Sands, but without any physical barrier in between, and the further 6 individuals from another dark soil population living a little closer, only 50 kilometers away, but behind the San Andres mountain range. So in that case, we have the barrier here. And one of the first things we were interested in was to describe the demographic history of the colonization event in the 2 species. And what you can see here is our best fit model for cow lazy. And what you can see is that the model that describes the data first is an admixture model whereby the white sand population was created by admixture of the 2 dark populations with a larger fraction or a larger proportion actually coming from the closer location, the one that's behind the mountain range here. But I would also like to draw your attention to 2 details in this model. The first is the time of admixture, which is estimated to be about 3,500 years ago, so just very shortly after the formation of white sands. And the other is that this model suggests that there is still ongoing gene flow with asymmetric migration between the demes, with a higher migration into white sands than out of it. In contrast to that, if we look in the case of Inornata, we see that here the white population is actually much more closely related to the other population within this Tularossa basin, so the one that is further away but without the physical barrier. But we also see that split times here are estimated to be much younger, only about 1,400 years ago, and in this model there's less to no evidence of gene flow between any of the populations. Apart from the inherent interest in characterizing the demographic history of colonization in white sand lizards, our demographic estimates are also important because they provide us with an important null model for tests of positive selection. So we took a composite likelihood approach to evaluate the likelihood of selection across the MC1R gene where we found the previous mutation in. And as you can see here, for the 2 dark mutations that I've shown you here in the 2 different blue colors, there was no significant test value found in any of the 2 species, whereas if we look in the 2 light mutations shown here in red, there were strong rejections of neutrality, suggesting that there, or at least giving evidence that there is selection in the planched populations in these MC1R gene. And interestingly, these likelihood surfaces are peaked or centered near our previously identified mutations, which I showed you here in the red bar. And at that point, I would like to remind you that they have a different genetic underlying architecture. So in this case, the mutation is recessive, and in this case, the mutation is dominant. And though the mean time to fixation should, in both cases, be similar, at least for large NNS, we expect a different pattern of a selective sweep. So if we think about the recessive case over here, the early allele frequency trajectory will be determined by genetic drift up until the recessive allele reaches a frequency by which it can appear in the homozygous recessive state, and at which time it will be seen by selection. Whereas in the dominant case, selection can act on the allele directly, but in later stages, the recessive allele can kind of hide out behind the dominant one in the heterozygous case or state. So later stages in the trajectory will be determined by drift. So from theoretical work that was done in 2006 by Molly Przewowski's lab, we know that these dynamics should result theoretically in a stronger reduction of diversity in the recessive case and a much more proda pattern in the dominant case, which is exactly what we're seeing here. So the signatures of selection at MC1R are confirming the inferred dominance, which I think is quite nicely done. Given the different colonization times of the 2 species, another thing we wanted to do is to estimate the allele ages of those beneficial alleles that were highlighted by our CLR test relative to the geological age of the white sand system. And for this, we used a newly proposed ABC method that will be published early next year. which takes into account the different dominance effects of the two mutations. And what I'm showing you here in the two plots are the joint posterior densities of the selection coefficient S and the allele HT in 4 and E generations, both on the log scale, for Inanata on the left and Kalesi on the right. The white, yellow, and red colors give you regions of high, moderate, and low density. And what you can hopefully see is that the inferred allele age is different between the two species. The allele is to be inferred much older in Kalesi than it is in Inanata, which is quite nicely confirming what we've seen from our demographic results. But most importantly, both sweeps are much younger than the geological age of the sandhills. So in summary, I think it is really interesting to see that different mutations in the same gene can act through different functional mechanisms, even if they result in the same or very similar phenotypes. Alleles can differ in their dominance and in their net selection coefficients, for instance, due to differences in pleiotropy or epistatic interactions, which then in turn can affect the probability and rate of fixations of these alleles in the wild. And part of my future research in collaboration with Pri Rosenblum's lab at Berkeley will focus on extending our knowledge about the cryptic coloration in wild lizards at wide sense by including a third lizard species, the lesser earless lizard, which you can see here, for which preliminary work suggested that the putatively beneficial mutation in the MC1R gene that is responsible for this blanched coloration doesn't change receptor signaling, unlike in the two species that we've just seen. So this species somehow has evolved a different way to change coloration with a very different mechanism, and that's, I think, really interesting for advancing our knowledge in this area. As I mentioned earlier, there are many examples for cryptic coloration in nature, and the second system I'm going to talk about is cryptic coloration in the deer mouse. In particularly in deer mouse populations that inhabit the ecologically distinct and recently formed sandhill system in Nebraska, which you can see in the background picture here, where, similar to the lizards inhabiting white sands, mice living on the sandhills have evolved cryptic lighter coat coloration, as you can see here, relative to their much darker relatives that live in the dark soil of the surrounding prairie. Previous research from the Hoekstra lab has identified mutations here in the agouti locus, not in MC1R, that are responsible for this color change. And this has greatly enhanced our understanding of the role of selection or the mechanisms driving it. But what we're still struggling with is to understand the role of selection on a genome-wide scale in wild populations, because their approach, like many others, use genome-wide scans to identify regions of selection. Whereas what we would ideally like to be able to do is to have a way to witness evolution happening in action. And one great way of achieving this is by conducting manipulative field experiments, which can help us to isolate specific evolutionary processes by controlling, for example, for demographic variability or by restricting gene flow. My collaborators Ron Barrett and Hopi Hoekstra spearheaded a project in which they constructed 6 really large-scale enclosures, which you can see in the bottom pictures here, 3 on the light soil, so on the sand dunes, and 3 on the dark soil, in order to directly measure the fitness consequences of pigmentation differences on both the phenotype and the genotype. So these enclosures are 4 fenced with really large steel plates. They're about 1.5 meters overground and about 60 centimeters underground. So they prevent the mice from burrowing and escaping. But other than that, they're really natural environments. So mice that live there are still subject to avian predation. And what they've done is they cleaned out these enclosures from all the living critters in there, and they seeded them with mice that they have collected both on and off the sandhills, so from both environments, hundreds of them. And each of the mice were labeled before, So over a time period of about 2 years, they came back every few months and trapped these mice in live traps and recorded which ones were still alive in order to monitor for shifts both in phenotypic trait distributions but also in genotype frequencies. And one of the first things we observed within the first 15 months of the experiment is that we've seen shifts in the phenotypic distribution of pigmentation traits. And I'm showing you here dorsal brightness just as one of the examples. And if you focus on the right side here for a moment, so this is one of the light enclosures, what you can see is that mice that survive, which are shown here in red, are much lighter than the previously colonizing population, which I show you here in blue. And the same is true vice versa for the dark side, indicating that cryptic mice are being favored or favorably survive in each of the two sites. So what we would ultimately like to know, as I just said, is what's the genomic consequence, what's the entire genomic response to this observed phenotypic selection. And in order to answer this question, we generated a genetic variant data set from all of our initial nearly 500 deer mice of the original colonizing population, and we have the record of which of the mice were surviving. So one way to characterize the genome-wide response to the observed phenotypic selection is by quantifying allele frequency change before and after an event of selection, with the main idea being here that you would expect to see a large allele frequency change at causal sites due to direct selection, but also at very closely linked sites due to indirect selection. There's obviously a problem because the deer mouse genome is really large, so for a lot of sites we might see large shifts in allele frequency simply due to chance, due to random genetic drift. So in order to distinguish the effects of natural selection and random genetic drift as the causes of allele frequency change within the single generation, we employed a method that consists of 3 different null models that was proposed by Gompert and his colleagues earlier last year, whereby the first null model tests whether the magnitude of allele frequency change at the locus is consistent with evolution by genetic drift. So for this, we used a Monte Carlo Bayesian method to obtain the distribution of the expected allele frequency change under the null hypothesis that survival and genotype were independent. And we were doing that by permutating our true survival vector, which I show you here as a square, several thousand times. And for each of the permutated survival vectors, we calculate the allele frequency change. This then gives us rise to a distribution to which we can compare our observed value to. Whenever this observed value falls in the tail of this distribution, we call it significant or a loci with exceptional allele frequency change. The second null model then tests whether the number of loci with exceptional allele frequency change that we observe is any different than what we would expect from genetic drift alone. And without going into any details, conditional under rejecting the 2 first models, we then have developed a third null model which tests whether one SNP under selection is sufficient to explain the observation that we have. And this is still very preliminary data, but I just wanted to give you an idea whether we think we need selection to explain the changes in allele frequency that we observe. So what I show you here are the results for only one of the enclosure pairs out of the 3 pairs that we have. And what I want you to focus on first is the agouti locus, which we already know is of interest from previous studies. And what I show you here in the red bar is the number of SNPs or loci with significant allele frequency changes, and then the 2 distributions that we obtain from null model 2 and 3. So the red one is the neutral one, and the blue one assumes that there is one SNP under selection. On the bottom is the result for the light enclosure, and what you can see here is that we reject neutrality, but one SNP under selection is actually sufficient to explain the observation that we have. Whereas in the dark environment, we reject both the neutral model and our model with one SNP under selection, suggesting there might be multiple SNPs under selection in the agouti locus here. We also had a look at the random regions of the genome, so everything but agouti, basically. In the dark case, we can't reject neutrality. In the case of the light enclosure, we again reject both, suggesting that there might be other regions in the genome outside of agouti that are under selection in these mice. As I said, this is still very preliminary work, and our next steps will focus on analyzing data jointly from all the 6 enclosures, which should give us much greater statistical power. But we're also working on extending our model to actually explore how many SNPs under selection are necessary to explain our observation. Additionally, we are in the process of generating cross-generational data for more than 650 progeny that we have in these enclosures from the original founders, which will also allow us to estimate selection on both viability and fertility. In order to identify and characterize other genetic determinants of local adaptation, we also have collected data from more than 500 deer mice together with corresponding soil samples along a linear transect in the sandhills. And transgenics are currently underway to validate some of the identified mutations, and results are looking extremely promising for that. So this gives us hopefully a better understanding of the system. With this, I would like to end my talk and acknowledge all my collaborators, Peter, Gil, and Molly for collaborating on the chimpanzee project, Magnus for collaborating on the Vervet monkeys, and Jeff, Bree, Rowan, and Hopi for collaborating on the cryptic coloration. And thank you for listening.
So we would like people to use the microphones.
Okay. Good talk. So one of the things in mutation research is sort of the new frontier in humans is the trying to get it done in nutritionally stressed humans. Sort of get away from city populations. So like the CEU is like Western population. The Yorubans like a city population. So get to more nutritionally stressed. Is anything going on the same in say recombination research?
I mean, probably not on the same scale. There has been, I think, in the last year or two, there have been studies published in African populations, which show recombination rates or hotspot locations which are very different to, say, Europeans. But I don't think it's on the same large scale. But I mean, I agree. I think it would be really important to look at more populations because we know that the mutational landscape is different between Europeans, Africans, people of Asian ancestry, and I think it would be great if we could include, I don't know, South American people and just get a better idea of what's going on.
So since I have the mic, I will continue. Fight me for it.
Can we see the hands so we can move the microphone?
So with recombination, so individuals that say had the SRY is translocated from the Y to the X, so they're XX males, they have sterility problems because meiosis doesn't properly function. Yeah,
I mean, that's true too. If you have a deletion in the pseudoautosomal region in humans, then males are sterile too, right?
And so is that, so at least for like XX males, right, the sperm should be used to having an X in it. Do you think that's more weird recombination occurring from meiosis, or you think it's just a failure because they're missing male fertility genes on the Y? Do you have any, does anybody think about just say how physical recombination issues in meiosis might affect?
I would think it's probably the latter, but I'm not sure.
So when you use population genetic data to estimate recombination rates, you're actually getting time averages
Yes.
of recombination.
Time and sex averages.
Do you have a sense as to what that time scale is and how it's weighted towards the present? Because if the recombination rates are changing over time, you might not actually be estimating recombination rates that have existed at any time.
That is true.
So do you know,
and
what's
a very
the
good
time
point.
scale?
I actually don't know.
I mean, that'll become,
Yeah, no.
it relates to your question as well. If
Yeah.
there are environmental effects, you're actually getting an environmental average, an average over the environmental states that the populations
That
were
is true.
exposed to.
I think especially since we know that at least within human populations, hotspot locations are changing very quickly by this self-destructive drive of the motif that we're seeing. Yeah, but I don't know which time scales we are looking at. I think it also depends on how closely your individuals are related you're looking at.
Mm-hmm. So, I mean, that could explain your chimp data, the fact that you're
That
not
is
finding
true.
a correspondence between the binding motifs for the protein and
Yeah.
the motifs that are there. It may be that the current state of the protein is actually affecting current recombination rates, but you can't
not,
discern
we
that
can't see
from
it.
the data.
Yeah, that might be true.
Did somebody else have their hand up? Oh, right here. Just keep asking.
Who wants?
I was joking.
I have a question. So, okay. So, what I find interesting with the recombination rate and the correlation with factors, I mean, early on when we were discussing it was the repetitive elements, right? We had these nice microsatellites at that locus, slippage repeats, and all these things which were clear mechanisms to increase recombination rates, which we understand. Then we moved to the larger scale, right? And then all these correlations went away, basically. And so I've never really believed that that single gene is explaining all the variation we see, right? And so I wonder whether these transposable elements, repetitive elements, which vary between those, explain a lot of the variance you see between chimpanzees and humans.
So I haven't been talking about it, but yes, we see definitely differences in those repetitive elements. Some of them seem to be attracting high recombination rates in chimps and not as much in humans and vice versa, but they're definitely important in both species. But they don't seem to be at the same level in the two.
Level in terms of recombination frequency variation?
Yes. Yeah, I mean, the level in terms of you might see a lot of hotspots in certain repeated regions in humans, but not necessarily the same repeat structure in chimpanzees, whereas other repeat elements might be less important in humans, but more turned on in chimpanzees.
But you see effects
But
of
we-
repetitive
yes, we do.
elements on recombination
Yeah.
on that micro scale.
Yeah.
Cool talk.
Thanks.
In mice, there's balancing selection. In chimps, there's balancing selection, long-term balancing selection. But in baboons, there's no long-term balancing selection. So I'm wondering how that falls out, like for monkeys, for
I
vert
think
monkeys.
I probably am most interested in things that we think might be important over the long evolutionary run, such as genes involved in immunity. So maybe, I don't know, vervet monkeys and different monkeys had similar pressures from viruses and kind of evolved similar mechanisms there. But also in terms of blood groups, we know that there's balancing selection going on in the ABO in humans and chimps. I think that would be interesting to look at too. Yeah.
I think we have time for perhaps one or two more.
Oh, Melissa.
You got 10?
I was curious in the survival of the light mice and the dark mice. Just looking at the table, it looked like the light mice always had higher mortality on every background.
Sorry.
Yeah, I guess it's, well, one, is that the case? And two, was there a test to look at survival just of light versus dark mice, regardless of the background?
So if you're looking at the different soil colors, which is what I'm showing you here, we see that mice that live in the light enclosures are actually killed much more frequently by birds, particularly owls, I think in our case, than mice that are placed in the dark environments, right? We have about 50% of the mice surviving and much less in the light environments. And I think part of the reason is the different environment they're living in. So the dark environment has a lot of grass, it has bushes where mice can hide in.
So this is total number of mice regardless
Exactly,
of their coat color.
regardless of the coat color. So these, exactly.
Okay.
So they were,
Sorry,
when they were initially introduced, in each of the enclosures, they seeded about half of the mice coming from a dark soil environment and half from a light. And they put all of them in the dark enclosures, and the same for the light. So all of the enclosures were seeded with half light, half dark mice.
And on the plot right before, did dark mice, the distribution there, it looks like there's
So-
a much lower survival of.
So, I mean, in principle, what they seeded with is in a range of half, in principle, half light and half dark mice, which is the original founders, which I show you here in blue. And then we have the two environments. So this is the dark soil environment, and this is the light soil environment. And the mice that are surviving in the light soil environment are on average lighter, and the mice that are surviving in the dark soil environment are on average darker, but here the effect is not as large. And as I was saying, I think it has something to do with how the environment looks like, because on the light soil, it's really just sandy and scrubby, and there are not many hiding places out there. So it seems like if you're a dark mouse that's placed in this light environment, you're really sticking out. Whereas if you're a light mouse in a dark environment, you can maybe hide under the bush and kind of escape the eye of the predator in a way.
So one question?
Yeah.
Last question.
So with your Vervet monkey data,
Yes.
so you said you had 160 individuals,
Yes.
whole genome. What's the depth? What's the quality? What else? I mean, if you have a lot of stuff, you could rely on that for years, doing
Yes.
all sorts of stuff.
Yeah,
with
I know.
that
That's what I
species.
was saying. I think I'm particularly interested in the recombination rate, but I think this can be used for a lot of different things. So coverage varies between low coverage of maybe 4 or 5x up until maybe 45 per individual. So we have kind of a whole range depending on how much DNA they could get from the individual, anything in that range. But we have a lot of high coverage individuals too in there. It's the 130 that are sampled from Africa are from all these 5 subtypes that I've shown you. And I mean, the 30 from the Caribbean are thought to come from that location in West Africa that were poured over.
And with that, we should wrap it up. Thanks, everybody, for joining us.
Thank you. Thank you.
That was great.
Thanks.